Now let's talk about another remarkable member of extracellular matrix. This molecule, fibronectin, has binding sites for different other molecules and also molecules that perform the function of cellular adhesion to extracellular matrix. Let's talk about fibronectin. Fibronectin is a large glycoprotein found in all vertebrates, and it contributes to organization of ECM and also helps cell attached to the extracellular matrix. Fibronectin is a dimer. It has two molecules which are linked together by the disulfide bonds. Each subunit is folded into series of functionally distinct domains. So here these different domains, and they're functionally distinct, and they're separated by regions of flexible polypeptide chain. For example, this is the flexible region, and you can think of it as a hinge. So this molecule is a flexible molecule. In fact, the flexibility of this molecule plays an important role in its function. We'll talk about that in just a few minutes. The two polypeptide chains are similar, but they are generally not identical. They are made from the same messenger RNA, which is spliced alternatively, generating two different forms of fibronectin. Here, I've already mentioned that the disulfide bonds holds these two units together. The C termini of each chain is joined by the two disulfide bonds near the C terminus. Each chain is 2500 amino acid long and is folded into 5 or 6 domains connected by flexible polypeptide segments. So here, as I've already pointed out, these are the flexible polypeptide fragments. Individual domains are responsible for have different functions. For example, this yellow dot here basically means it's the yellow domain is basically self-association domain, where fibronectin molecules can associate with each other, they can bind with each other. This is, for example, the domain which allows fibronectin to bind or interact with collagen molecule. Here, the red domain shown here in red is basically the domain where cells can bind via, for example, integrin molecules. And this blue domain shown here is basically the area where heparin can bind. The domains, in turn, consist of smaller modules. The domains we have shown, we saw that yellow dot and blue dot and the red dot, these are made up of smaller modules, each of which is serially repeated and usually encoded by a separate axon. This basically suggests that fibronectin gene, like collagen gene, may have evolved by multiple axon duplications. So the axons of collagen are roughly either 54 nucleotides long, or multiple of 54 nucleotides. So this information suggests that this probably occurred due to gene duplication. All forms of fibronectin are encoded by a single large gene, and this gene contains about 50 axons of similar size. The transcription, which produces RNA molecule, is alternatively spliced and to generate different isoforms of fibronectin. The main type of module is called type 3 fibronectin repeat, and this basically has 90 amino acids, and it occurs at least 50 times in each subunit. So here, the red domain has the smaller modules shown here, right here. We have said that they are type 3 fibronectin repeat. The type 3 fibronectin repeat is one of the most common of all protein domains in vertebrate. Type 3 repeat is the main repeating module in fibronectin. It also has a special sequence, RGD, basically these, which are symbols for these 3 amino acids. And this is the region which basically binds, or rather integrin binds to this particular sequence of amino acids. It also has a synergy domain, the module. The synergy domain is also cell binding site. Short peptides containing RGD sequence can compete with fibronectin for binding site on cells and inhibit the attachment of the cells to fibronectin matrix. If these peptides are glued onto a surface, a solid surface, they can cause the cells to adhere to this RGD sequence, which we said were the 3 amino acids. And this is also found in a number of other extracellular proteins, for example, blood clotting factor fibrinogen. Fibrinogen proteins contain this RGD sequence, and they have been useful in development of anti-clogging drugs that mimic these peptides. Snakes use a similar strategy to cause their victims to bleed. They secrete RG-containing anticoagulating proteins called disintegrants into their venom. So the reason why certain people bleed excessively when they're bitten by a snake is because presence of this sequence in the snake venom. RGD sequence are recognized by several integrins. We have seen that different integrins are dimers of alpha and beta subunits, and there's several types of alpha and beta subunits forming different integrins. And each integrin specifically recognizes its own small set of matrix molecules. So just binding RGD sequence is not enough. These integrin molecules also need additional sequences that allow them to bind those sequences more strongly. Here you can see is an electron micrograph of fibronectin molecule. The red arrow is pointing at the location where there is the disulfide bridge. And fibronectin molecules, very important components of our extracellular matrix. We'll talk some more about them in the next module.